morning, everyone, and welcome back. This is session 9, BioMarine 2015, Drugs from the Deep Ocean Bioprospecting. My name is Rory Francis, Executive Director of the Prince Edward Island BioAlliance in Eastern Canada. And it's a great privilege to be here with our distinguished panel this morning to talk about this important topic. We have with us on my right, first, John Coleman, Vice President and Scientific Director for RC Inpharma in Florida, United States. We also have Catherine Brisson, Chief Operating Officer of Stellar Biotechnologies, California, USA. And far right, Torfinn Hansen, who is Chairman of the Board, Arctic Biodiscovery Development Center in Tromsø, Norway. So welcome to all our panel. We have about a half hour to discuss this topic, and I want to first start by asking each of our panelists for a brief introduction of themselves, what their focus is in their business, and I'll start first on my right here with John.
Yes, my name is John Coleman. I'm a co-owner and scientific officer of RC Inpharma. We're located in Doral, Florida, the gateway to the Caribbean and the gateway to South America. I work with Professor Luis Romero, who's a specialist in dengue fever and chikungunya, which are predominantly infesting the entire Caribbean and South America right now in waves. We're developing dengue, chikungunya therapeutics because we find that probably half the patients who come into the hospitals there are co-infected with dengue and chikungunya fever virus, which is a mosquito-borne virus spreading rampantly through Brazil, rampantly through most of the tropical countries. We've found a potent antiviral inhibitor of dengue 2, the big killer, with a combination of 2 algae extracts from cold water algae. We have another compound that's a land plant. It's an alkaloid being used to treat another disease, so we know a lot about the pharmacology of that thing. And we find they're very powerful potentiators of each other, and together they are knocking out dengue 2 at very low submicromolar, nanomolar levels in human cells. We are ready to proceed with our clinical trial in Santo Domingo at a hospital that specializes in nothing but dengue fever. They have an entire ward that's dengue fever and chikungunya fever dedicated, and believe me, they're crowded. The average cost to treat dengue fever in most of these South American countries is about $1,800 a week before they can release them back into the wild. Some of these small countries is bankrupting their economy essentially. With the new wave of chikungunya on top of the dengue, it will bankrupt those countries. It's a WHO priority. It's a priority of the U.S. Army and the U.S. military because many of these soldiers sent overseas in these hot areas are also getting dengue and chikungunya infections. Most of you have not heard of chikungunya, but it's a lot worse than dengue fever, let me tell you. Dengue fever, you're flat on your back for 6 weeks and then 3 months in a malaise. When the mother of the house or the father of the house is stricken with dengue at the same time the kids are, it's really a bad situation. We had a family of 5 in Peru. The entire family died from a dengue 2 variant. Dengue 2 is becoming more virulent. It showed up in Brazil, it showed up in Peru, it showed up in Ecuador and in Venezuela. We're having 50% mortality in adults. This is a world crisis situation. Approximately 50% of the world's population lives in tropical, semi-tropical areas, and they are all being infested by it. Southeast Asia very badly. Brazil has the largest population of dengue survivors. Dengue infections today, about 70% of all South America, the infections are in Brazil. But that's only the people who are making it to the hospitals and the clinics. We don't have records on those who stay in their villages. So that's where we are right now. We've done our in vitro work. We're ready for a clinical trial at Robert Carval Hospital, and that's where we are. We need a 300 to 400 person clinical trial under FDA and pharma standards, double-blind, placebo-controlled.
So hold that thought, because we'll come back to some of that commercialization pathway part, John, momentarily. So, Catherine.
Hi, I'm Catherine Brisson. I'm with Stellar Biotechnologies. I'm the Chief Operating Officer. We are a commercial stage biomarine company specializing in the production of keyhole limpet hemocyanin. This is a very important immune stimulating protein used as a carrier protein in these immunotherapies and as immunodiagnostic for diagnosing immune competency. It is sourced from one animal, and that's a marine mollusk that's native only to the California and northern Baja Peninsula coastline.
Thanks, Catherine.
Yeah,
Torfinn.
and my name is Torfinn Hansen. My academic background is in biochemistry. My professional background is from the pharmaceutical industry. Many years as CEO for Wyeth in Norway and Denmark. After the merge with Pfizer, the CEO for Pfizer in Norway. Nowadays, my life is mainly dedicated to transfer bioprospecting into business, and mainly through my work with Arctic Biodiscovery Center.
Thanks very much, Torfinn. So you all obviously are working in marine natural products, marine products as your source of technology to bring to market to solve some of the larger challenges of human health and other applications. But let's go back to maybe the beginning of where these technologies are coming from. So Catherine, let's start with you. The limpet is sort of your engine for producing a high-value compound. Where did that technology come from originally, and perhaps where did the bioprospecting happen that led your company to commercialize that technology?
Well, the founder of our company, Frank Oakes, is really the initiator of the aquaculture program, developing that technology, developing the manufacturing process. It really dates back to nearly 100 years ago when some of the first papers were written on the anatomy of the giant keyhole limpet, the Megathura crenulata. And so since then, into the 30s, 40s, and 50s, it was really considered the immunologist tool, primary tool for diagnosing immune competency. In the early 90s, a biotech company in Canada was working on a bladder cancer drug that was using KLH, and they really had no idea where it came from. They assumed it was a reagent that they just ordered from a catalog and had no idea what the source of the material was. So in understanding what their dilemma was, we worked on putting together culturing the animal, spawning the animal on land, raising it up through the larval, post-larval stages, all the way to an adult, where we non-lethally extract the protein and purify it.
Torfinn, can you, in your experience in Tromsø and through maybe your Pfizer previous experience, talk about a similar example of where you folks are working on commercialization of a particular marine source bioactive and kind of where that process is at the moment?
Yeah. Up to now, we have only a few examples of drugs derived from the ocean or the sea. It should be many, many more, and that's make the opportunities when we are doing prospecting based on marine species. And our concept is that species living in cold water, 1,000 to 2,000 meters of depth, in extreme tough conditions, they have, through thousands of years, developed survival mechanism with structure which may have, or is expected to have, biological efficacy or effects in humans. And that's a concept for the research which is being done. And based on that, we have collected a lot of species from the sea, and then the work is to characterize and then test it in different models in order to detect biological efficacy, and then based on hits, explore whether this could be used in as ingredients, pharmaceuticals, nutraceuticals, etc. And we are always aiming for the highest level in the bioeconomy value pyramid.
John, your source of material that you're working on for dengue fever and other diseases is an algae source. You said a couple of species of algae. So I'm
yes.
wondering, where did that technology come from? Did your company identify those algae strains and do the preliminary work to identify efficacy, or what was the source of that material?
There are many other sulfated polysaccharides from brown and red seaweed that have been known for several decades to have antiviral activity. They first looked at HIV-1, then they looked at herpes simplex 1 through 6, and finally, when dengue really became a problem 5 or 6 years ago, they began exploring the possibilities that these sulfated polysaccharides would work on dengue. The problem is the molecular size of these sulfated polysaccharides are sometimes 60 kilodaltons to 100 kilodaltons. They will not penetrate the intestinal membrane or the cell membrane, so they'll clear the virions and neutralize the viral transmitter particles called virions in the bloodstream, but they won't get into the cells. But what we found is 5,000 to 10,000 kilodaltons will penetrate cell membranes, will pass the blood-brain barrier, and be useful in treating dengue in human beings.
So the particular question of source, did these strains, were they identified and selected through assays and so on by your company, or did you partner with an academic research laboratory who identified these? Trying to get back to the source of where did these
We did
come
a 25,000 screening of 25,000 compounds, and we have 25 that are major hits.
That sounds like a lot of work.
A lot of work.
25,000. So talk about that a little bit. Is that what kind of process did you use to screen from 25,000 down to 25?
Well, we were doing in vitro studies. We were doing, thanks to flow cytometry now, we can do 96 samples at a time instead of one. Operator doing one sample at a time. And we got it down to 25 major compounds that have had very big hits against dengue, 1, 2, 3, and 4.
Mm-hmm. So I'm going to ask you all to comment on marine. We're talking about marine sources here. So why marine? It just happened to be where that chemistry lie that was important to you, or is there something about marine prospects and chemistry that provides a particular advantage, perhaps in terms of solving some of these big challenges? Torfinn?
I think for us it's obvious that the closeness to the Arctic Sea is the main background for taking care of this part of the research. And added to that, what I already mentioned, that we expect to find a lot of candidates for a lot of the diseases that still is a demand in the market for obvious treatment. But it's the closeness to the Arctic Sea and the Arctic area which make it unique to do this research in Tromsø and also other Norwegian research institutions.
Catherine, you want to speak to that?
Well, for Stellar, it was really about KLH and identifying a way to provide a sustainable, scalable source. It was had already been identified as a go-to tool in immunology, and it was then going forward in immunotherapy. It was about how to get the body to recognize, to present antigens from an immunotherapy standpoint. And since the keyhole limpet is not really identified as a food source, that was the primary role for us, and making sure that we could develop a GMP grade pharmaceutical KLH for immunotherapies.
John, what do you think about the ocean marine sources of solutions to some of these diseases that you folks are working on versus other possibilities?
Yeah, there are land plants, but the marine sources, the sulfated polysaccharides, are the only source that we've ever found. We've found nothing like that in land plants. We found alkaloids, we find steroids, we find saponins, but nothing that has the activity that the algae, microalgae have, and the sulfated polysaccharides from seaweeds.
So just thinking for a moment outside of your own companies and the focus you have now, When you think about the marine sources of new chemistry and new solutions, what about biodiversity? Do you feel that generally there's been a fairly complete exploration, as some would argue there has been in terms of land plants and some what's available there? Have we gone as far with marine chemistry and marine bioprospecting to identify some very interesting new solutions and new technologies? Torfinn?
As already mentioned from this theme, I think we are only in the beginning of the track in order to explore all the possibilities within the ocean. And in Tromsø, this has been a history for only 20-25 years, but after the establishment of this center in Tromsø, we have learned that there is a lot of interest in other countries, specifically from other countries in Europe, which for years has been focused on substitute from land, that they really are exhausted of the start now as we are in the beginning of exploring also the species from the sea.
Catherine?
Well, as a commercial stage biomarine company specializing in KLH, I really can only speak to KLH, but we're continuously looking for new uses, new opportunities to utilize KLH, and we really feel like there's some opportunities still out there.
So, Catherine, in your case, I mean, the other question that arises when we're talking about marine sources of biomass and technologies is sustainability of supply. So it's fine, we find something interesting. Do we have to carve up a reef off Australia in order to get, that enough security of supply for a company to have a commercial business, or are there other alternatives to that in terms of ensuring that there is a way of both taking advantage of the technology but doing it in a sustainable way? Catherine, your company, obviously, you're all about a particular model for production. Maybe talk a bit more about that. And ask John a similar question. You're looking at algae sources. How do you ensure that's a sustainable system? I'll come back to Torfinn. So, Catherine, maybe start with you on.
Sure. We at Stellar, we maintain a first-in-class on-land aquaculture system. We spawn the animals. We actually can induce the spawning process. We do the fertilization. We raise them up through the larval and post-larval stages, all done with animals generation after generation, in which have been spawned at our facility. We non-lethally, non-harmfully extract the protein from this animal several times a year. We return them to give them a nice sushi grade dinner and get them back into a nice recovery tank. And then we do all the manufacturing of the product, the GMP grade KLH at our facility. So it has taken years of research in order to understand the life cycle of this animal. We have patented technology related to the extraction of the hemolymph from the animal. So it's really a culmination of over a decade of work in order to develop these proprietary techniques to ensure that we can sustain the animal and make sure that we can continue to scale up to meet the demands of KLH and the current immunotherapies.
So, John, in your case with algae, assuming you're early, so you're still going to clinical trials and a lot of questions to be answered, but assuming an algae source is what you may be working with as in a commercial sense, pretty straightforward. It's algae, it's microalgae, so you can scale that up as required. Any limitations, any sustainability considerations that?
No, not really, because the Philippines have a great model of growing seaweed on these long wooden poles they put under the low tide limit, and they cut off a 2-foot piece of seaweed and they attach it to the pole, and then in 6 months they harvest a 6-foot or 8-foot long section of seaweed. So they have entire farms they're using for some commercialized sulfated seaweeds that are used in food drug use.
Mm-hmm. Torfinn?
I don't think sustainability is really relevant or so relevant as we are talking about drugs. It's more relevant for other uses of marine biomass, which in the end normally is sold through farming. The reason why it's not so relevant for drugs is that I think the main route is to characterize the molecule and then make it synthetic. And then you have solved the problem or the challenge of sustainability.
Can I just interject there?
Absolutely.
We have spent years trying to understand the KLH molecule, and many times it's tried to be synthesized unsuccessfully. It's a very highly glycosylated molecule, and as such, all the attempts have really stripped off the immunogenic effect. So in our case, sustainability and having a productive aquaculture system is required.
You have the similar problem today, for instance, in biological drugs, and there the solution is farming or maturing through a bacteria or so on.
So a quick advertisement for one of the companies in our cluster, Nautilus Biosciences, which has a bank of microbes, bacterial and fungal microbes from various parts of the world, dereplicated collection. And what they've found over their research period is that it's, for example, not the coral that's producing that interesting chemistry. It's actually the bacterial populations that are living in that coral. So therefore, you don't really need to harvest coral. You can harvest those bacterial cultures if you know how to do that and sustain them and reproduce them. And then the scale up is actually not so challenging because you can do it in a controlled environment as opposed to a continuous supply from the ocean. So what about commercialization and moving from kind of the prospecting side to commercialization of these marine products? Are there particular challenges that are associated with the marine side of things, or at that point this is just chemistry and therefore it's not that different from other sources when it comes to commercializing the technology? Anything specific, or what are some of the challenges that you folks see in the commercialization pathway? So, John?
Well, yes, the sulfated polysaccharides from seaweeds are very poorly characterized, as much as people like to think that they are well characterized. They go from anywhere from 10% to 35% sulfation, and the cold water species have a higher percentage of sulfation than the warm water species. But they also have higher molecular weight, which makes that a problem. So just even understanding the structures of these polysaccharides is very challenging. And it's being done, but it's going to take years to really characterize exactly what's going on. Catherine?
You folks are well advanced, obviously. You've got commercial products, but just in general, any thoughts?
Sure. For Stellar, it tends to be dual focused. The regulatory interaction, we have to adhere to the US FDA requirements along with other international regulatory bodies, the US EPA, the California EPA, the US Navy, the California Coastal Commission, Fish and Wildlife of California and the US. So we adhere to many, many different regulatory bodies. And the other part is, because we work in the immunotherapy and the aquaculture marine biology side, is really having a diverse expertise in both areas.
Mm-hmm. Yeah, Torfinn?
Yeah, and added to those challenges, you have a lot of challenges in general when developing drug. And I think the main obstacle is financing, because it's enormous cost today to develop a drug in the order of $2 billion US dollar, for instance. And there also been a shift in the dynamics concerning development of drugs. 10, 20 years ago, most new NCA was developed through big pharma. Nowadays, it's totally changed. Now it's startup companies which make the basic research, and then they are partnering with big pharma in order to not only get financing but also competences to fulfill the development and also the international commercialization. So just succeed in partnering with resources with necessary finance muscles and competence is a challenge in general, specifically for products needed to the regulatory challenge already mentioned.
So we just have a couple of minutes left to wind up. Maybe I'll just ask you each for your views in general about the marine side of prospecting for new technologies, new solutions in human animal health nutrition. And Torfinn, let's start with you and come across.
I think one important point is, of course, you should aim for being in the top part of the value pyramid concerning bioeconomy. But based on the obstacles specifically for marine products and also in general, I also think that one alternative route could be to go for advanced nutraceuticals. That is, you have to do some clinical trials confirming the mode of action and the concept, and going that route could end up to have substance with a position like a drug, but you have a faster route to the market and also an easier route normally to the market. But then you have other IP challenges and so on, which is more specific if you succeed to obtain the drug status.
Catherine, any last points you want to make?
Well, for Stellar and having achieved a sustainable supply manufacture of KLH, we just hope that future bioprocessing or bioprospecting from the ocean would make sure that it's looking from the sustainable point of view going forward, because we all have to be shepherds of the ocean.
Thanks, Catherine. John?
Yes, our products are fully sustainable and can be grown in any of these countries that have a coastline. They can farm the algae and create a cottage industry of extracting the compounds that we really need. And that in itself will give these countries another little small economic boost. We found one thing, though, that the average cost of improving a drug in the United States is $900 million. If 1 out of 10 drugs are finally approved, that's $9 billion. The shortcut is to go through the FDA botanical drug claims, which is 1,000 times less regulatory if it's a natural compound. So we're going that route simultaneously with doing clinical trials in the Dominican Republic where there are no rules.
Yeah. Well, thank you very much, everyone. Fascinating discussion, and really appreciate your time and contribution to the conference. Thank you.
Thank you.